Hello guys, I am Shubham from Flow Tutorials. We are continuing with the site-specific recombination. So before we start, subscribe to our channel and press the bell icon to get notified whenever new video is out. So in the previous video, we have seen the two examples of the tyrosine recombinase, that is integrase and phase 3. In this video, we will discuss the other two remaining tyrosine family recombinases, that is Yeast FLP and E.coli Xer CD. Okay, so what it does basically, first of all, we will see Xer CD, right? So what it does, promotes several DNA deletion reactions, okay, that convert dimeric circular DNA molecules into monomers. Means dimers are converted into monomers, and thus recognition sites are two sites are there. First is the plasmid bond site CER and chromosomal sites CIF. Okay, so these are the two sites for recognition of these recombinases. So let's see with the images now. See, this is a dimer, basically conserves site-specific recombination. A DNA replication has occurred, homologous recombination, right? These are the crossover points. And in the final, what we see, this is the dimer, see here. So these are the dimers. Now this has to be separated. So this separation is done by site-specific recombination by the enzyme XERCN, right? So these enzymes perform this function and the dimers are now separated into monomers, chromosome monomers, right? So as we saw, this is an example of tyrosine recombinase family. So here will be the formation of holiday junction intermediate, right? First stand cleavage, then it's joining, then holiday junction formation, and then second stand cleavage. Okay, so this is done by the basically two enzymes as we saw, XCRC and XCRD. This is the heterotetramer. Okay, tetramer means 4 subunits are there. Okay, so 2 subunits of XCRC, right, and 2 subunits of XCRD. Okay, so this is the heterotetramer structure, right? So what happens here? FTSK, there is additional protein known as FTSK, which do the activity, important role, okay, of site-specific recombination. How? Let's see first of all FTSK independent pathway. What happens here? Only XCRC is active, right? Only this protein is active, XCRC. But when FTSK is there, so this is FTSK, okay, then only the D is active, okay, then D will do the first strand cleavage, right, and then the XCRC will be activated. Okay, so here independent pathway, this is an incomplete pathway. Okay, this is incomplete and this is complete, right? So this is the difference. So FTSK will make this both the proteins active, T as well as C. In the independent pathway, only C is active, right? So this is that much only of XCRC, which is important one. Let's summarize about XCRC and D. So what it does, dimers to monomers, right? And with the help of FTSK. right? Then in FTSK, independent only XCRC active, and independent one both XCRC and D are active. Okay, so that's only we have to know about XCRC and D. Now the next enzyme we will see Yeast FLP. Okay, XCRC and D are present in E. coli. Do note, this is also important thing. E. coli, and this FLP is present in yeast, right? FLP is basically flip. We can say flip. So what it does, it inverts, okay, inversion of a region in the yeast to microplasmid to allow for DNA amplification reaction called rolling circle replication. So that's only we have to know. And the recombination sites are called FRT sites. Okay, flip, recombinase, target, right? Recognition site we can say, where FLP will go and bind. As we saw in the Cre recombinase, it was used in what we can say, to produce the gene knockouts. As well as this FLP FRT system is also used in Drosophila. Okay, Drosophila, it does mitotic crossing over and recombination, which give rise to, okay, to create homologous mutations, right, and conditional knockouts, right? So let's see one example here with the help of diagram. So this is a heterozygous, okay, cell, Drosophila cell. This chromosome is from maternal one and paternal chromosome. Paternal chromosome is having normal gene X, and maternal chromosome is mutant, right? Mutant. And both these chromosomes are having FRT sites where FLP will go and bind, right? So this is that is only written here. This is homozygous for FRT and heterozygous for mutant X, right? And here chromosome replication will be take. After replication, what happens? The chromosome is duplicated, and then we are inducing the signal, activate FLP gene promoter. So when this is activated, FLP protein will be formed, which is a recombinase, right? And this will do the recombination thing. See, catalyze, mitotic crossing over, and recombination. That's only the thing here. And after crossing over, does the, what we can say, as we know, crossing over, what is exchange of genetic material, exchange of strands. So that has occurred, and the cell will divide further. After division, what has happened? One cell is with homozygous mutant and one cell is with homozygous normal gene. That's it. And after proliferation, the clonal proliferation gives homozygous patches in wings, right? So this is how we can produce the homozygous mutants. Let's summarize the yeast FLP now. That's only we have to know about these things. Not much. Yeast FLP. What it does? Inversion. Okay, inversion, rolling circle replication, rolling circle application, as well as mitotic recombination giving rise to homozygous mutants and conditional knockouts. So this FLP FRT system is used for this thing, for reduction of conditional knockouts and homozygous mutants in Drosophila, right? With the help of mitotic recombination. So that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section.